Hello, everyone, and welcome to EAStalk webinar series. My name is Anna Veskovic, and I'm the Program and Project Manager at the European Aquaculture Society, and I will be your host today. We are really glad to have you with us for what promises to be a fascinating session on cutting-edge health monitoring in aquaculture. Before we get started, a few quick housekeeping notes. Today's webinar is being recorded, and the recording will be shared with all the registered participants in the following days. So don't worry if you need to step away or want to revisit anything. For our EAS members, the presentation slides will be available to download for free in the members area after the webinar. If you have any questions, please use the Q&A function. We will be having a dedicated Q&A session for 15 minutes at the end of the presentation. I will be keeping an eye on the questions as they come in, so feel free to submit it at any point during the talk. Today's talk sits at the heart of a really pressing challenge. As the global demand for animal protein grows and climate pressures mount, aquaculture needs a smarter and more welfare-focused solutions. The Ignition project is tackling this heads-on, exploring molecular signatures in minimal invasive matrix like skin mucus, and translating those findings into electrochemical biosensors for real-time health monitoring. We will hear about work across European seabass, Atlantic salmon, and rainbow trout, so plenty to get into. Now let me introduce our speakers for today's. Cláudia Magalhães is a PhD researcher and junior scientific researcher at Sparos and Research in Portugal. Cláudia's expertise sits at the intersection of molecular sciences, multiomics, bioinformatics, and biostatics, with a strong focus on non-invasive biomarker strategies, particularly through the fish-skin mucus interface. Her work aims to identify accurate signatures of fish performance and welfare in aquaculture systems. Our second presenter is Luiza Wasiewska. She is a Research Ireland-funded postdoctoral fellow and a senior researcher at Tyndall National Institute in Ireland. Luiza combines microbiology and biosensors expertise to develop next-generation diagnostic tool. Her current research explores alternatives to antibody-based recognition molecules, with application ranging from bacterial detection to stress and inflammation monitoring. And she's also actively researching sustainable materials like bioplastic for use in electrochemical sensors. So together, today, Cláudia and Luiza will be presenting their work on harnessing novel non-invasive biomarkers for biosensor-based health monitoring in aquaculture, insights from the EU Ignition projects. Over to you, Cláudia.
All right. Can you hear me properly?
Yes.
Yeah. All right. So hello, everyone. Thank you for the introduction, Anna. So I will skip that part because you gave a really complete introduction already. So I'm here to talk about this part of this big European project that is called Ignition, and I will talk about this component of the biomarkers part. In this presentation that is entitled Harnessing Novel Non-Invasive Biomarkers for Biosensor-Based Health Monitoring in Aquaculture, Insights from the EU Ignition project. So just a brief introduction to this thematic of ignition. So we all know that there is a high demand to deliver high quality protein worldwide. And we know that aquaculture is part of the answer for this. So because of that, there is an increased pressure on the aquaculture sector, while at the same time, we need to pay attention to the environment, sustainability, and to ethical issues. And of course, we know that if there's this increased pressure in the production, also there's an increased pressure on animals. And regarding that, fish increase their stress exposure and then also consequently diseases spreading. And now we have an additional challenges regarding the climate change because it further exacerbates the pressure on animal welfare. And it is precisely on these last 2 points that the Ignition project is focused on. So in this presentation, I will focus on 3 of the main objectives of the project. Okay, so objective number 1 is to study animal welfare. and we will try to increase our knowledge on the molecular, physiological, and behavioral responses of different species in the context of climate change. The second one is the biomarkers discovery, as the title said. So we are identifying health and stress biomarkers on minimally invasive biological matrices through high-throughput omics screenings coupled with bioinformatics. And finally, the last but not least, the biosensing part, which will then be presented by Luiza, where we are implementing innovative biosensors to detect these minimally invasive biomarkers that were identified before, and with this to monitor animal health continuously in a non-invasive or minimal invasive way. So as I was saying, we divided this presentation into parts. So I will talk about the multiomics analysis that we did during ignition and the biomarkers identification. And then Luiza will present us the final product of the utilization of these biomarkers, which will be the biosensors. So the multiomics, I don't know if you're all familiarized with it, but omics is the study of the whole sets of biomolecules in an organism, a tissue or a sample. And so multiomics comprises several of these omics. So for example, transcriptomics regards the study of gene expression. Proteomics regards the study of all the proteins in a sample. Metabolomics, all the metabolites involved also in the organism, and so on. So multiomics concerns the integration of all these several layers of regulation. And with this, we can have a systems level overview of the organism when it is responding to a challenge or a stimuli. It can be the diet, stress, disease, and this will help us to track how the change in a gene, the expression of a gene, will lead then further to physiological changes and also a phenotype. So this infographics actually summarizes very well the work we've been doing in this work package of the biomarkers. So we will integrate different data that comes from different sources. So we will have, and we analyze already the classical biomarkers that we use to typically assess, like cortisol, glucose, lactate, and other physiological indicators. We are working on novel biomarkers, and this is the focus of this presentation, in minimally invasive biological matrices, such as the skin and the skin mucus, and using these high-throughput omics technologies, and also operational welfare indicators, and these can be changes in behavior. And we also have a partner in Ignition working on this that is working in machine vision. And then we will integrate all this data into a database, and through machine learning and predictive models, we will identify these novel biomarkers and associate them with specific endpoints, okay? And this will allow for quicker actions that will accelerate disease detection and then improve fish health. So why did we choose the skin mucus? Why working on the skin mucus? So this biofluid is actually very rich and very informative for a biomarker identification. So the skin mucus, it's the outermost layer that protects the skin, and it's a multifunctional shield against the external environment. So it works as a physical defense, as chemical defense, and it is part of the innate immune defense of the fish. Also, it reflects the physiological states of the animal. So it's the composition of the mucus actually changes with stress or with an immune insult. So it changes not only the viscosity of the mucus, but also the biomolecules that are in it. And it allows to measure different types of molecules, so stress hormones, like cortisol, for example, immune indicators, molecular indicators. And also very important is that it's a non-lethal source of information. So we can repeatedly take measures, of course, with distance. And as I was saying, there are a lot of secreted biomolecules in this biofluid because there are a lot of extracellular components from not only the skin cells, but also from internal organs. And we actually want these proteins or these biomolecules that are secreted from the cells into the mucus because it facilitates then the monitoring with the biosensors and hopefully with reduced pre-processing, so without lots of steps of extraction to achieve those molecules, to reach those molecules. And so for the biomarker identification, we need to work on two approaches and an integrative approach, which is, it's important to have this mathematical approach for the identification of the biomarkers using the predictive models, but also we need to pay attention to the functional biology, because we want these biomolecules to have biological functions that are, of course, involved in the pathways that we want, that are important for stress and for health. And so we were working on first a data-driven approach, which identifies novel candidates through mathematical optimization and large-scale pattern recognition. Then with a knowledge-driven approach that leverages the biological plausibility to guide interpretation and hypothesis generation. Then ideally, we will work with an integrative approach that will bridge discovery with physiological context and ensure the credibility of our identified features and prevent the overfitting of our models. So I will show you here a short video that summarizes all this work that we've been doing. Okay, so now let's jump into the experimental trials that we conducted at the Ignition. So as Anna mentioned, so we are working with 3 species, the European seabass, the rainbow trout, and the Atlantic salmon. And we exposed these different species to different trials and to different challenging conditions. So on the left, you see that we did a transport trial, and transport trial was to simulate an acute stress. And the fish were sampled then pre-stress, so before the stress was applied, immediately after stress, and after a 24-hour recovery period. And then for the temperature trials, which simulated a chronic stress, fish were exposed to a control temperature that was optimal for the species, a medium high temperature, and also a high temperature. And there was also another trial conducted with vaccination to assess the stressful event of the procedure itself. But we are working on this data still. Then different samples were taken. So we collected transcriptomics from the skin, proteomics from the skin mucus. The skin mucus was also used for the microRNAs. So those microRNAs are the small RNAs that regulate gene expression, and also blood and blood plasma for physiological parameters. And these samples were all collected the same for all the species and all the trials. So then these same challenges were repeated, again, the same trials, but this time with a dietary condition. And so we tested diets supplemented with 2 halophyte extracts that were very rich in polyphenols at different inclusion levels. And these halophytes are actually very rich they display high antioxidant and anti-inflammatory properties. And they were developed by the Refine, which is a company in Denmark that is also a partner of this consortium. So then into the methodology. We optimized different omics pipelines in-house here at Sparus. And don't worry, because I will not go through all these softwares. Just wanted to mention that, for example, for the transcriptomics, we identified 53,000 genes for all the species in the trials. For the proteomics, we identified around 9,200 proteins, and for the microRNAs, around 320 known microRNAs and 77 potential novel microRNAs. And so all this information was then integrated using different supervised and unsupervised models. And I will give you here the example of MOFA. So MOFA is an unsupervised approach, and it's a multiomics factor analysis. And it works more or less like a principal component analysis. So instead of our variables here, instead of the single features, they are our different data sets of omics and other data that, as you will see. So here, for this case, for example, this is an example of seabass. We integrated all the data from the different trials and of the different omics, and also the physiological data from the blood plasma. And here on the left, you see that the numbers of features that were integrated into the model for the seabass in this case. And so now the results. This will not be too exhaustive because it's a lot of molecular data. So it's just a brief overview of all the data that we obtained and highlight some results. So in this table, I tried to summarize all the differential features of the different species, the different omics and the different trials. So I don't want you to retain the numbers, of course. What I wanted to highlight here is for you to pay attention to the differences in the tissues and also the trials. So we see that in the skin, we had a stronger response immediately after stress. So the skin responded immediately after the stress, and we see the highest number of differential features here. But on the contrary, in the skin mucus, we see a stronger response after 24 hours. So you see, comparing with the last group, that we have a highest number of differential features here compared to stress. Okay, this for the acute stress. And then for the temperature, as expected, we had a higher number of differential features for the different omics with in responding to the highest temperature group. And for the salmon, we are still working on the data for the temperature trial. And then for this presentation, I focused on the common features because we want, of course, biomarkers of stress, but we also want species specific. So we are looking for both of types of biomarkers. And for the common features, it's important to remind that these trials were conducted in different countries, in different systems, and we found common features between all of them. So they are all responding with these markers. So for the transcriptomics, for example, we found 40 common features in the skin between all the species. And most important is that they were not only identified, but they were also share the same regulation between all these data sets, as you see here in this heat map. And I will not go through all of them, of course. I just wanted to highlight a few ones that were identified with a very promising involvement in the stress response, but also they were identified by the machine learning model as the ones of the most important for the model. So for example, this is one, FKBP5. It's an important modulator of the stress response, and it actually modulates the glucocorticoid receptors. The DTIT4, it's a stress-responsive gene, and it plays an important role in cellular energy and cellular stress, and also in response to epoxy and DNA damage. FOXO3b, sorry, is a transcription factor that regulates apoptosis and the oxidative stress and tissue remodeling. The TX-nipase regulates oxidative stress and glucose metabolism, and it acts as an oxidative stress mediator. And finally, ZF and 5A, there is not much information about this one, but it's involved in protein degradation and stress responses. And all these are very promising as candidate biomarkers as well, because they were also identified in the skin mucus at the protein level. Okay, so this is exactly what we want. We want to follow the whole flow of information from the skin to the skin mucus, and we want preferably, as I said initially, these proteins to be secreted in the mucus, so to facilitate, of course, the monitoring by the biosensors. Then regarding the proteomics, we had 11 common features between all the different species. And just to highlight a few ones, the R2, it's a metabolic enzyme that is induced by epoxy and oxidative stress. MMP9 is an extracellular matrix enzyme that is also involved in leukocyte migration, tissue repair, and mucosal barrier remodeling. And CFH, it's a key regulator of the complement activation that modulates the innate immune response. So we see that in the proteomics, which is in the skin mucus layer, we have a response that is more directed to the innate immune defense. And regarding the microRNAs, we have 3 common ones, which is the miR100, which is the mTOR regulated. mTOR is a pathway that is a master regulator of protein biosynthesis, and it's involved in cell growth, autophagy, and cellular stress. We have the miR363, response to infection and stress, and also regulates apoptosis and inflammation. And also miR221, which is involved in the response to oxidative stress, in the immune response, and inflammatory regulation. So the microRNA response was actually very promising because it was the one that was more consistent between all the trials and all the species. And these 2 microRNAs specifically, there are already studies published with aquatic organisms identifying them as responding to different Vibrio diseases and also stress. So then regarding the multiomics integration, with this model, we can select, or the model selects a set of features that are classified, let's say, as the most important for the model, the ones that have the highest weight on the different factors. And these features that are selected for the model, I'm giving here the example for the seabass. In this case, it's selected 10 for transcriptomics, 10 proteins, 5 microRNAs, and 4 physiological indicators, will integrate a composite stress score. And it's called composite because there are different features in it. And with this composite stress score, we can also generate an equation that, using the expression values of these different biomolecules, will be used to calculate a stress score for our samples. And I will show you here the example. So using the training samples, so the samples that were used to generate the model, and using those features that I mentioned before, the model actually classified very well the samples into the control group and the stress group. Of course, this has to be further validated using blind samples, so samples that the model has never seen, to validate this stress score. And with this new trial that I mentioned with the allophytes, we want to assess if the stress score decreases in the groups fed the allophyte supplemented diets. So the groups that were stressed and were fed the allophyte diets, we want to use this approach to see if this score decreases. And this will be one of the steps of the validation. So this composite stress score can be used on this biomarker signature, can be used in a multi-analyte assay. So you can simultaneously monitor different features. And these kind of multi-analyte biosensors already exist, especially for humans. And for example, this study that I mentioned here, it uses already protein and nucleic acids. So it's something we are working on. And now I will pass to Luiza.
Hello, everyone. It's my pleasure to be here and tell you more about the work we are doing here in Tyndall as part of Ignition. So just as a introduction first, what are the biosensors? Biosensors are analytical devices for rapid detection of target analytes. So they compose of two main elements, which is biorecognition element and transducer. So biorecognition element, there are different types. There are biomolecules like antibodies, nucleic acids, aptamers, enzymes, which recognize specifically different analyte in complex sample. And basically this recognition element, for example, if antibody binds with a target protein, this recognition element can be then transduced using either electrochemical, optical, mechanical techniques into measurable signal. So basically in our lab, we mostly use electrochemical biosensors, which transduce this biorecognition event into a measurable electrochemical current. And then we can detect this signal and process the results. So the main advantages of this method is that it's very sensitive, because you can detect literally single electrons, so it's way more sensitive than, for example, optical methods. It's rapid. The measurement can be done within seconds or minutes. You can use small sample size like microliters. It has suitability for miniaturization and point of care devices, and also can be scalable. So here is the example. Actually, not only we are working on electrochemical sensors, but we specifically focus on printed electrochemical sensors, which can be printed on using our benchtop printer. And this basically removes the need for clean rooms, which are very expensive, and a lot of processes to make the sensors is very expensive and requires several steps. In here, we need one single step, unless we use different inks, maybe then you need to change the ink. And on top of using the printed sensors, I'm also very interested in sustainability, because usually this kind of sensors are unfortunately single use, because also of the problem of biocontamination. As you can think of a COVID test, a lot of COVID tests after being used, there were millions during COVID that were then thrown in the bin. So we are trying to make them more sustainable, that eventually they can actually be composted and don't persist in the environment. So here is a quick video of how the printer works and how it's printing sensor. So as you can see, it takes less than a minute to print single sensor, so we can really scale this process. And also the design can be changed very easily without the need for specific masks. And so you can make different size of the working electrode, which will influence then the sensitivity of the sensor, and introducing multiplex electrodes. So for now, this is the example of dual sensor, but there is also a possibility for introducing more electrodes, and then you can detect more analytes at the same time. So at the moment, we are focusing on detection of IL-1 beta, which is a stress marker, because we started this work before knowing all the exciting results from Cláudia. So this is the example of immunosensor, where we actually use the antibodies for detection. So the quick scheme of like how we develop the sensor: we start with the bare electrode, we modify it with a polymer to introduce the functional groups to which you can attach in this case antibodies. So we use a lot chitosan because it has it is very abundant in amine groups, which then you can attach the antibodies to. Then we have our analyte recognition, and there are several different techniques in electrochemistry which you can use. We often use impedance measurement, which measures the resistance of the electrode, and the more analyte attach, the more resistance increase, so the signal increase. And the challenge is to optimize the sensors for to work the way we want to, with high sensitivity and selectivity. So here's some example of the steps we need to optimize the sensor. For example, as I said, chitosan contains a lot of amine groups, and here we use the dye which attach specifically to the amine groups, and we want the most homogeneous coverage of the electrode. And here in the, you can see the example in the picture B, where the yellow color is really homogeneous on all electrodes. We also need to increase sensitivity with this, for example, the chitosan is not conductive polymer, so we need to add some nanomaterials that I will show you later to increase the sensitivity. Biomolecule concentration is important, and of course at the end, if you want to work in complex samples, the selectivity is very important. So here is one example of the results that we usually get. So the signal change with increased concentration of the target analyte. Here we actually focus just on the lower range of this concentration in the further. And here, as I said, we only modified the electrode with chitosan, and what we definitely saw the change with increased concentration, but we were not happy with how big the change was. So it was below 20% change even in the highest concentrations. So then we introduced another layer of gold nanoparticles between the electrode and the polymer to increase the sensitivity. And as you can see, the response increased even up to 40% signal change. Then the important part is to actually test the selectivity. Here we use interleukin-6 as a negative control. And unfortunately, we saw some response, which was less than the target, but still the problem is if you have unknown sample, if the signal increase, you don't know if it's because it's a low concentration of target or it's unspecific. So when we used the electron microscopy, you could see that because of introducing this layer of gold nanomaterials, there was only this patch of polymer, so the polymer wasn't deposited that homogeneously anymore. So we changed this, we optimized again the polymer deposition, and as you can see here, it's very nicely, then the response between the positive and negative was very different. And again, going back to the electron microscopy, you can see that the polymer was distributed more homogeneously. So this is the work for now with detection of the stress markers of the protein. And we are also working now on the microRNA detection. So the detection is in some ways similar. We use the same way of attachment of the probe nucleic acid. And then after the hybridization, we use methylene blue, which is a molecule that intercalates between the double-strand nucleic acid, and we can detect that again with electrochemistry. So I had the master student now finishing that was working on that. Here you can see just few graphs where the single-strand probe, the signal didn't change after PBS. Then with very small concentration of microRNA still didn't change, but then started increasing with increased concentrations. And we are looking again at optimization of this detection. So again, using increasing number of the nanomaterial, gold nanostructures. So here there are 3, 5, 8 cycles of, so you can see actually the detection with 3 and 5 cycles is quite good with good linearity. But then the more cycles, it actually doesn't improve it anymore. So as you can see, just the challenge of the steps that need to be taken to optimize this detection. And yeah, so our next steps is to finalize the sensor for microRNA detection and also to confirm that its selectivity. And then we will move to testing real samples, as Cláudia was mentioning, mucus, but also skin. And ideally, we would like to detect these biomarkers in fish tank water, but the levels may be very, very small. But then it would be the really non-invasive way. But we also will work with the mucus and introduce the multiplex detection. Thank you. Back to Cláudia.
Cláudia, you're muted.
Yeah, sorry, just noticed. So just wanted to share with you that as part of the dissemination of the project, we also developed a few games that are available at this page that you can see here. Regarding different thematics of the ignition. And for example, we also developed one that is here, the photos, regarding these parts of the biomarkers and the biosensors. I think it's very interesting, and it's available for download as well. And we have also different resources such as infographics and also videos and fact sheets. Okay. And this is the consortium of the project. And that's it. So thank you all for your attention. And if you have any questions, just put it in the Q&A.
Thank you so much, Cláudia and Luiza. This was a fascinating overview of where the field is heading. The idea of being able to monitor fish health continuously and non-invasively in real time has a huge implications for the whole sector. So we are now opening the Q&A. So I would like to remind you all that please send us your questions. We have 15 minutes for the Q&A. I will start with the first one to Cláudia. Will you be testing these biomarkers in a large production settings?
Yeah, actually, so the next step will be to do this in a commercial scale. So we are working with also a partner of Ignition, which is Gifish. It's a company that produces salmon in Norway. And we are already at Sparos, we are developing the diets now with these alophyte extracts, actually the ones that were selected as most promising. And we will soon, I think next month, we will conduct this trial there in Gifish, and we will test and then analyze the same biomarkers and assess if we can have the same regulation.
Thank you. The next question is beyond the molecular responses, did you also look at the fish physiology, and were you able to draw any correlations there?
Yeah, so we assessed different physiological parameters in the blood and the blood plasma, such as the cortisol, glucose, lactate, also some immune parameters as well. And it was actually very interesting that, for example, as I mentioned, and if you remember in the MOFA model, so the Multiomics factor analysis model, we can also include other data other than omics data. And we also included this physiological data. And then it finds correlations between the different features. So the omics features, all the omics features and the physiological ones. And there was a strong correlation, for example, with the cortisol, both in the mucus and in the plasma. Actually, I didn't show that data, but yeah, there was a strong correlation with the cortisol for the acute stress as expected.
Thank you. The next question I have is for Luiza. Please submit any questions that you might be having for Cláudia and Luiza. Don't be shy, and now is the time to ask the question. So the question that I have for Luiza is, what are the main challenges of commercializing electrochemical biosensors?
The main challenges we are facing is reproducibility, actually, because especially when you start, as I showed you, there are many steps that you have to modify the electrodes, the biomolecules, which again, moving to biology, things are never as reproducible as you can, as you would think from the engineering point of view. So the more steps you introduce, the more things can change in the process. So the reproducibility is a big issue. And when product needs to be commercial, it has to be really reproducible. And then with the printed sensors as well, the process is way faster and easier, but it can't really follow there is a reason for this clean room microfabrications. So we are trying to make it as reproducible as possible. And then also the stability, for example, of the biomolecules, antibodies, so you want to avoid the cold chain, but then the biomolecules can disintegrate. So there's still a lot of challenges in the field that we are working on.
Thank you, Luiza. We just received a question from Paul Sindralau. How far do you think you're from a commercial product?
For the biosensors?
I believe so.
I believe so.
Yeah, so it depends what commercial products. We have actually in the group a commercialization project now. But it's for a different application. So we hope that actually the plan is even next year or two, but it's with a different type of sensor, so it's not the printed sensors. But I think I would say it depends also on the target. So the biosensors are a bit more challenging, as I said, because you need the biomolecules, but there are other ways with only chemical sensing, which would maybe be a bit easier to enter the market. So I think within few years, less than 5 years, I would say we will see definitely more electrochemical sensors on the market.
Thank you, Luiza. And can the printed sensor be fully compostable now?
So what I was showing you, we printed on the bioplastic, which is actually commercial bioplastic, which can be composted in the industrial settings. But the challenge is the inks, because we still use the gold inks. Because the gold is just the most conductive material. So that is the challenge, to move away to different kinds of inks. They're carbon. So I submitted another proposal for working on that. That needs to be changed either to carbon, or I see actually a lot about zinc, which is supposedly also could be used as a compostable material. So that is the challenge, to get away actually from the gold, and from gold, silver, platinum. Usually it's the noble metals, which are used because of their properties, which are excellent for electrochemistry. But yeah, we need to get away from that to make it actually compostable.
Thank you, Luiza. Cláudia, I have a question now for you. It's about halophyte extracts specifically. So how were these chosen, and what inclusion rates were used for this?
So we have another partner, which is Nord in Norway, that is also working on that. So there were several in vitro trials that were conducted. If I correctly remember, we've had kidney leukocytes, and also with gill epithelial cell lines of salmon and of seabass. I'm not sure about the trout. And these preliminary in vitro trials were the ones that were also used to select the different inclusion rates, and also the different extracts. Because we had, for example, I presented here, I said 2, in the material method, but before we had more, and we have more inclusion rates. And then we narrowed down to these 2 with these preliminary trials.
Thank you, Claudia. We just received a question from Arasidian Gaze. Thank you for a nice presentation. How many samples per group is good enough for confidence in omics analysis?
That's very depending on your context and of course on the effect size of the features that you are analyzing. There's not a standard number of samples that it's good enough for any omics or the condition that you want to test. Of course, it depends on the budgets. You need to consider a lot of factors. And also on the variability of the data that you are analyzing. So it's always important to have these preliminary studies to analyze that variability before you conduct big trials to know the exact number of samples for the response that you are assessing. I cannot say like an ideal number of samples for each omics.
Thank you, Claudia. We just received a question from Ariadna Sitnia. Have you already tried a sensor with fish mucus for interleukin 1 beta?
This is our next step, actually. So that's because we were working on making it selective, as I was showing you. So with this problem, we are like, okay, we need to work on that. And now it's actually, this is our next step. So watch this space.
Thank you, Luiza. As we discussed just prior about commercialization of the electrochemical biosensors, I'm interested to know about the molecular markers. Do you think, Claudia, if they could reach aquaculture companies in the near term, or is it more relevant to experimental research for now?
Yeah, it's without a doubt, it's very important for experimental research because especially in this context of climate change, that we want to analyze the effects of the different factors, temperature, pH, and the other factors in other conditions in the fish. So we want to know the effects at long term that they have on the animals. It's without a doubt very important. But of course, we also want to reach commercial farms, and that's our main objective. And I think, like Luiza was saying, I cannot precise like a window for the next years to commercialize this. But I know, for example, there are a lot of types of other intradermal sensors, for example, that are already commercialized that detect other types of indicators. And so, yeah, it's our aim as well to reach that commercialization in the near future.
Thank you so much. Regarding the next phase as well, what does the path look for translating these biomarker signatures into operational biosensors tools for industry use, in your opinion?
So the next steps for the biomarkers.
Yes.
So this, yeah, there are a lot of steps still of validation. We are at this lab validation stage now. But for example, we need to test this, as I was saying, on blind samples with from new trials to achieve this the best performance for the models, of course, and have the most precise biomarkers identified. And then we also need to be able to identify them using other techniques, less sensible techniques, because we did all these high throughput omics for now as a screening. But then when we want to detect these biomarkers in the future, when they are already validated, we want to detect them with more rapid, faster techniques and less sensible, for example, PCR or an ELISA. And so they need this change, the full change of these features need to be detectable by those techniques. Another thing that needs to be optimized is the threshold value. So we need to, for the biosensors, we need to be able to say that above a certain value or below a certain value, the fish as a compromised stress or health. So it's also something that we are working on. And yeah, I think that's it. And further more technical steps involving the biosensors, of course.
And how do you envision this as a handheld device that farmers could use, or how they will read all these information that they receive from biosensors?
There is a lot of work done in the area to miniaturize, because at the moment in the lab we use big potentiostats, but there are already commercially available small, like very, very small potentiostats, and also we have great engineers in the group who work on miniaturization, and then apps. Like we live in the era of apps, and there are already apps that then can be connected to your phone, collect the data. So this field is going very fast in this direction, miniaturization, and yeah, then also introducing AI to analyze the data. So this is going very fast.
Thank you, Luiza. The next question we got is from Jabed Hassan. Could you please share your experience regarding the stability of these biosensors under varying and environmental conditions such as temperature, salinity, and pH?
So yeah, we didn't do this tests yet because we are at the validation stage. But we have data from previous sensors in the group as well that could definitely the sensors can survive at least a few weeks in dry conditions. But yeah, we usually test them in the fridge, so that is another challenge to actually have them without the cold chain, because as we know, the biomolecules will always be more stable in the cold. So yeah, that's the next step to validate that. But also I'm very interested in actually not using antibodies in the future. There is the field also is going in the direction of alternative molecules to antibodies, so more synthetic antibodies that could then also be in line with this challenge to make them more stable in different temperatures and maybe pH. And so actually my other research is focusing on that. In this project we use antibodies, but I think it's a very promising area as well to use more stable molecules in the future.
And does sample handling or the storage affect the results that you get?
Yes, that will be the fact in biology in general. I would say that how I think where we want to go is to actually analyze the samples immediately without storing, because normally it has to be minus 80 degrees, which is also increase the cost of. So ideally you can just test it immediately. And the sample processing can be a bit tricky, especially with the skin, with the mucus, which is very, very, very thick kind of sample. So maybe it will need to be a bit diluted in some kind of buffer. But we've seen that already with COVID tests. You get, the buffer with your tests. So I would see something like that. And I think it will need to be at least a bit diluted in the buffer.
Thank you so much, Luiza. As we are coming to the end of the Q&A, I would like to remind you all that, we are coming to the end of the EAStalk webinar. As you maybe realize that Cláudia and Luiza left their email addresses there. So if you feel like emailing them with for more information about the projects that they are involved in, please feel free to send them an email. But I have a last question for Luiza, and that is, what are the next steps in your sensor research? to conclude today's webinar?
So yeah, as I mentioned, actually, answering to some of the questions, I'm very interested in making them fully sustainable. So developing inks that will be compostable, but still will have this properties that will allow us high sensitivity and the alternatives to antibodies. That's my big interest, because and I think that is, yeah, the antibodies will, we need something that is more stable and can survive longer and in different conditions. So
Cláudia, do you have any last words to say on your end as well?
No, I don't think so. I think it was a great discussion. Thank you, Anna.
Thank you so much to both of you, and thanks to everybody who joined us today. The webinar recording will be sent out to all of you by the end of this week or beginning of next week. If you want to explore the Ignition project further, please, you can visit their website at ignition/project.eu. I also shared the link to their website here in the Q&A. Thanks again for joining us, and it's a pleasure from the EAS part to be in the consortium of this quite important project. Thank you.